Good day. My name is Karel Svoboda. I'm a group leader at Geneva Farm Research Campus of the Howard Hughes Medical Institute. And in the first lecture, I introduced a whole bunch of powerful imaging approaches to study and track the function and plasticity of individual synapses. And now I'd like to show you how we can apply these tools to study individual synapses, calcium signaling in individual synapses, and really focus on the signaling pathways downstream of calcium that determine the synapse specificity and the spatial interactions between synapses. I've shown you movies like these where synapses are activated in a calcium imaging experiment, and you can see that NMDA receptor-mediated calcium influx is limited to individual dendritic spines, and these kinds of images teach us that synaptic transmission is point-to-point, and that the calcium signals downstream of NMDA receptor activation are synapse-specific. This suggests, of course, that the induction of synaptic plasticity might also be synapse-specific. That is, if we activate this synapse strongly to induce synaptic plasticity, we see synaptic plasticity only at this synapse, but not at neighboring synapses. 2-photon glutamate uncaging allows us to address and show this directly, to address this point directly. Here is the experiment. We use 2-photon glutamate uncaging to induce long-term potentiation, or LTP, of synaptic transmission. We first measure baseline amplitude of synaptic current in response to glutamate uncaging. Then we induce plasticity and measure the amplitude of the response to uncaging glutamate thereafter. And if we induce glutamate uncaging, if we induce plasticity at one synapse, we probe the current before and after at that synapse, but also at neighboring synapses with well-defined spatial relationships to the potentiated synapse. We call these nearby synapses. They're sort of in the range of 1 to 4 micrometers apart, so in very small neighborhoods of the dendritic branch. And what you can see here is that if you induce long-term potentiation in one synapse, you get, in fact, changes in the postsynaptic sensitivity to glutamate, but no changes in the postsynaptic sensitivity to glutamate in nearby synapses. Here are the population data. So the potentiated synapses changed by about a factor of 2. So the spine becomes twice as sensitive to glutamate uncaging. This is likely due to the insertion of AMPA receptors into the postsynaptic dendritic spine, but there's no change in the sensitivity to glutamate uncaging in nearby spines. So that tells us that long-term potentiation is input-specific at the level of individual dendritic spines. Notice something else interesting in this experiment is that the potentiated dendritic spine becomes larger. You can see that because this GFP-expressing neuron, the spine in this GFP-expressing neuron becomes brighter, and the brightness of this dendritic spine is proportional to the spine head volume. And if we now look at this spine enlargement, which is associated with LTP, we find that the spine enlargement too is limited to the dendritic spine that receives the LTP stimulus, the potentiated spine, and is completely absent from nearby spines. You can go one step further and correlate the spine enlargement with the change in the postsynaptic current at the level of individual dendritic spines. You can see that there's an excellent correlation. The larger the amplitude of the LTP, change of the postsynaptic current, the larger the change in the spine volume. So let me summarize these points. So I've shown you that synaptic transmission is point-to-point, and that the calcium signals coming in to dendritic spines in response to NMDA receptor activation are synapse-specific. They're limited to individual dendritic spines. We also understand the mechanism of how calcium is limited to individual dendritic spine, which might seem surprising because calcium is a highly diffusible, a rapidly diffusible small ion. But what happens is that calcium comes in through NMDA receptors, and it's rapidly extruded through the spine head membrane by highly specialized calcium extrusion mechanism. Calcium simply doesn't have time to dribble out through that narrow constrictive spine neck into the parent dendrite and out to invade neighboring spines. And I've also shown you that when we use long-term potentiation in individual dendritic spines, the effects of the inducing stimulus is limited to the spine that receives the LTP stimulus, so LTP and spine enlargement also synapse-specific. We can really conclude that synapses can function as independent units of memory storage. Now, it's not calcium. Calcium is absolutely necessary for the induction of long-term plasticity, and NMDA receptors are absolutely necessary. And in fact, it's the calcium coming in through NMDA receptors that are critical for the induction of LTP. But it's not calcium that changes synaptic function. It's biochemical pathways, signal transduction pathways downstream of calcium. In particular, the CaMKII and the RAS-MAP kinase pathways I've already described. We can now look at the function of these pathways under in LTP experiments using by measuring protein-protein interactions and FLIM microscopy. Here is a CaMKII sensor that was developed by Ryohei Yasuda's lab. CaMKII, when it's inactive, is folded on top of each other. And what Ryohei Yasuda's lab did is they tagged CaMKII on one terminus with GFP and on the other terminus with a dark YFP. And so there's fret when CaMKII is inactive, and you get a short fluorescence lifetime when CaMKII is inactive, and no fret when CaMKII becomes active. This sensor can now be expressed in brain slices, in a sparse subset of neurons in brain slices. Again, in these kinds of images, there are lots of dendrites in the field of view, but only one neuron is labeled, expresses the CaMKII sensor, and then we can combine fluorescence lifetime imaging using the CaMKII sensor with the induction of long-term potentiation at the level of individual synapses to ask what happens to CaMKII activation when LTP in use, and in particular whether or not CaMKII activation is limited to the spine that receives the LTP stimulus. So let me roll this movie now. This is the dendritic spine that receives the LTP stimulus, and we'll show that movie again here under baseline condition. Now the LTP stimulus, you can see you get a brief transient of CaMKII activity. Redder, hotter colors means more CaMKII activity, and subsequent to this burst of CaMKII activity induced by the LTP stimulus, you see this typical spine enlargement associated with long-term potentiation. You can also see pretty clearly that CaMKII activity is limited to the stimulated dendritic spine, to the spine that received the stimulus. When we look at population data, what we find is that indeed that although LTP and spine enlargement are synapse-specific, only the spine enlargement only occurs in the stimulated spine, the spine that receives the LTP stimulus. CaMKII activity is also limited to the activated dendritic spine, and the CaMKII transient is really quite rapid and really precedes the spine enlargement. So we think that the localization of the CaMKII transient after LTP induction, the localization of CaMKII activation after LTP induction likely underlies synapse specificity in LTP. How about RAS, the other canonical pathway? RAS, there are many good reasons to study RAS in the context of synaptic plasticity. RAS, as I already mentioned, is critical for the induction of long-term potentiation and spine enlargement. It's been shown by many groups. I won't show you that data here. And in neurons, RAS MAP kinase pathway is downstream of NMDA receptor activation and calcium. And RAS and its modulators, the GEFs, that activate RAS, and the gaps that inactivate RAS are highly concentrated in spine. I should say that RAS is a small GTPase that sits in the membrane. When it's inactive, it's bound to GDP, and then it's activated by exchanging GDP with GTP, catalyzed by GEFs, and activated RAS binds to RAF, which then activates MAP kinase and has many downstream effects. And so, and notice also that the interaction between RAF and RAS nicely amenable to measurement by FRET, because what we'll do is we'll tag RAF with one fluorescent protein, RAS with another fluorescent protein to construct a FRET sensor for RAS that is optimized for FLIM. And there are other reasons that RAS is of particular interest, because mutations in the RAS pathway are implicated in various forms of mental retardation. So here is the sensor that we developed to measure RAS activity. We call it FRAS-F, and we tag RAS with EGFP, and the RAS binding domain of RAF with a pair of red fluorescent proteins. And this sensor was really optimized for fluorescence lifetime imaging microscopy, and also tuned through many control experiments to be compatible with biology, to not perturb RAS signaling itself and not to really perturb the function of the neurons that express the RAS sensor. And we also made sure that the sensor actually reports RAS activity with good kinetics. In other words, that the fluorescence signals we measure with fluorescence lifetime imaging microscopy that report changes in the RAS sensor really very nicely correspond to the dynamics of endogenous RAS in the cell. So again, we can play the same game, express the RAS sensor in small numbers of neurons in a brain slice, and then induce LTP in individual dendritic spines and see what happens to RAS activity in spine enlargement after induction of LTP at the level of individual dendritic spines. I'll show you a movie where each frame is now 1 minute apart. We can get away with that because it turns out that RAS activity is slower than CaMKII activity. This is the spine which we induce long-term potentiation. Now you can see hotter colors corresponding to increased RAS activity, and also the spine enlargement that is associated with long-term potentiation as before. And we roll this again. RAS activity increases in the dendritic spine, but notice that in contrast to CaMKII activity, the increased RAS activity is not limited to the spine that receives the LTP stimulus. Rather, RAS activity escapes the activated dendritic spine and spreads through the dendrite and invades neighboring dendritic spines. In fact, RAS activity spreads roughly over 10 micrometers of dendrite, which encompasses about 10 to 20 synapses in one of these neurons. This is a quantification at the level of populations of experiments. Up here, RAS activity in the stimulated spine in black, and in the nearest neighbor spine in red. And down here, again, LTP. As before, LTP and spine enlargement are limited to the dendritic spine that receive the LTP stimulus, but RAS is not. RAS activity, activated RAS escapes the activated spine and invades neighboring spines to produce rather large changes in RAS activation over about 10 micrometers of dendrite. We can look at the spatial temporal dynamics of RAS in images like these, where we produce long-term potentiation in one spine and then look at the spatial profile of RAS activity as a function of time. Again, we find even at the level of populations of experiments that RAS spreads about over 10 micrometers of dendrite, and that RAS activity lasts about 5 minutes. So this is roughly the time and length scales. 10 micrometers, RAS active RAS spreads about 10 micrometers before it's inactivated, and it also remains active about 5 minutes. Using modeling approaches that I don't have time to go through with you here, we are pretty sure of what the mechanism is of the spread of RAS, and it's really quite simple. We think what happens is that RAS is activated in the activated dendritic spine, in the spine that receives the LTP stimulus, and then active RAS molecules escape the dendritic spine simply by diffusion, and then are activated by gaps, by the inactivating protein, which are distributed relatively homogeneously throughout the dendritic arbor. So it's just passive spread of activated RAS, which accounts for the spread of RAS activity after LTP induction. So we tentatively conclude that RAS signaling might not be synapse-specific, but might spread to neighboring synapses. And that conclusion is tentative because there are some potential artifacts in these kinds of measurements. I'd like to just go through with them with you briefly. And we call these uncertainty principles in the sense that any kind of measurement with an intracellular sensor of signal transduction also perturbs the system in rather precise ways. So for example, the RAS sensor that we designed is based on RAS itself, and then by introducing the RAS sensor in the neurons, we overexpress RAS, and overexpression of RAS could really lead to an artifactual increase in the spread of RAS by two mechanisms. One of them is that more RAS means that maybe the gaps, the neurons, the molecules that inactivate RAS are now less able to keep up with activated RAS because there are too many RAS molecules. So the expression of RAS might overcome or saturate the gaps, and then therefore RAS activity might be longer, and active RAS might spread over longer distances before being inactivated. Similarly, RAS might normally be bound by immobile or slowly mobile scaffolds, and again, the overexpression of the RAS sensor might overcome and saturate these scaffolds, leading to, again, artifactually increased mobility of RAS in these neurons. So the way we chose to control for these kinds of factors is to keep track of expression of the sensor very precisely, and measure the mobility of RAS and the inactivation time of RAS and the spatial spread of RAS activity, all as a function of RAS expression level. And RAS expression level was measured using immunofluorescence techniques that I don't have time to show you. And here is, for example, the measurement of RAS mobility, which was measured using RAS tagged with photoactivatable GFP. And this time constant is simply the time constant it takes, the time it takes for RAS to escape a dendritic spine. And you see that that's really quite independent of RAS expression level. That means that we're not really saturating some kind of RAS scaffold. And even if you extrapolate to 0 RAS overexpression level, you can see that you get sort of that this retention time of RAS in dendritic spines is sort of on the order of 4 seconds, and really independent to first order of RAS expression level. We can also look at the time scale over which RAS is activated, and again, that tends to be a small function of weak function of the RAS expression level. But even if we extrapolate to 0 RAS expression level, we find that RAS is still active for several minutes and isn't really strongly perturbed by expression level. And if we now directly look at the spread of RAS activity as a function of RAS expression level, we find that, again, that's at most a weak function of the concentration of the RAS sensor. And therefore, we can conclude perhaps a little bit more confidently now that RAS activity is really not synapse-specific, that RAS activated at individual dendritic spine in fact spreads over 10 micrometers of dendrite and invades about 20 synapses. So now the question might be, and it's somewhat paradoxical, how can a small ion like calcium be synapse-specific and be limited to these small microcompartments, even though ions are highly diffusible, while a membrane-bound protein like RAS, which is much more sluggish and more slowly diffusing, how can that escape a dendritic spine? Well, what matters here are two time scales. The retention time, the time over which the signaling factor, calcium and RAS, remain trapped in a dendritic spine before they can escape into the dendritic shaft and on to other spines. And the extrusion time for calcium, or the inactivation time for RAS. And the resolution of this riddle is that the extrusion time for calcium is incredibly fast. A calcium ion comes into the cytoplasm and is extruded 10 milliseconds later, while the retention time for calcium is much longer than that, hundreds of milliseconds. And therefore, calcium signaling, calcium signals, calcium ion signals are synapse-specific. In contrast, for RAS, the inactivation time is on the order of minutes, whereas the retention time is on the order of seconds. Therefore, active RAS has a chance to escape the spine that receives the LTP stimulus before being inactivated. For CaMKII, the inactivation time is about 30 seconds, and the retention time about 10 minutes. For that reason, CaMKII is also synapse-specific. So what I've shown you is that RAS signaling is not synapse-specific, that when we use LTP at individual dendritic spines, RAS is activated, synapse is potentiated and enlarged, but the RAS signals are not trapped in the activated dendritic spine, but actually travel to neighboring spines over about 10 micrometers of dendrite and involvings of 10 to 20 synapses. So neighboring synapses see a little bit attenuated and slightly time-delayed sort of puff of RAS activity. In contrast, CaMKII activity is synapse-specific, and it's really the synapse-specificity of CaMKII activity which likely underlies synapse-specificity in LTP. Now, what could be the biological meaning of this spatial spread of RAS? What are the consequences of the spatial spread of RAS? Well, inspired by previous experiments that have shown that interactions between synapses when weak stimuli are given in one set of synapses and strong LTP-inducing stimuli on another set of synapses, we did similar kinds of experiments, but now with micrometer spatial control. We had to again check synapse specificity in LTP because these experiments are done on slightly different conditions than the ones I've shown you a few slides ago. But under these conditions, again, LTP is induced at one synapse and has absolutely, the LTP induction has absolutely no effect on nearest neighbor synapses, just 1 or 2 micrometers away. So induction of LTP and spine enlargement, again, are input specific. Now, what if we tune down the stimulus to be subthreshold to LTP induction? So here, for example, we reduce the amount of NMDA receptor activation by a factor of 5. And that can be done in various ways, either by reducing the number of pulses with which we activate NMDA receptors, or the number of NMDA receptors activated per pulse. In either way, we now only get a transient spine enlargement which decays down to baseline, so no long-term potentiation. I should say that under these conditions, NMDA receptor activation is so weak that no RAS is also activated in the spine that receives this kind of weak stimulus. We call this a weak stimulus. Now, what if we pair a weak stimulus on one spine with a strong stimulus in another spine in a neighboring synapse? And this is the experiment. We first give a strong stimulus at time 0, and then sometimes later, in this case 90 seconds, we provide a strong a weak LTP stimulus, subthreshold stimulus, this kind of stimulus, to a neighboring spine a couple of microns away along the same dendrite. And you can see that after inducing LTP on one spine, we can now, with the weak stimulus, produce long-term potentiation at the weak spine. So what this means is that induction of plasticity at one dendritic spine reduces the threshold for inducing plasticity at a neighboring spine over some dendritic neighborhood, at least a few micrometers or so. Now, does this have anything to do with the spread of RAS? You could imagine that RAS spreads from the spine that receives the strong stimulus to the spine that receives the weak stimulus, now provides the missing RAS, the RAS that cannot be activated by the weak stimulus, and now allowing the weak stimulus to trigger a long-term potentiation in the neighboring spine. To test whether or not RAS might in fact be involved, we first looked at the spatial and temporal scale of the coupling between synapses. We call this also crosstalk, and compare that to the spatial and temporal scales of RAS signaling. So we varied the distance between the spine that received the strong stimulus and the spine received the weak stimulus, and the time between the strong and the weak stimulus. And what we find is that the spatial scale of coupling is about 10 micrometers. Here at 0 is the spine that received the strong stimulus. You can see you can go about 5 microns on either side of the strong stimulus and still allow a weak stimulus to evoke long-term potentiation. And that corresponds very nicely to the spatial spread of RAS activity, illustrated here by this green line. And similarly, if we wait about 5 minutes, we can wait up to about 5 minutes between the strong and weak stimulus to still allow the weak stimulus to produce solid, robust potentiation. And again, so the temporal scale of crosstalk between neighboring synapses matches very nicely to the temporal duration of RAS activity in these CA1 dendrites. Now, these are all still correlations, of course. We need to do a perturbation experiment to really prove that RAS MAP kinase pathway has anything to do with the coupling between synapses with reduction of the induction threshold for LTP in this dendritic neighborhood. So we came up with the following experiment. We reasoned that, well, maybe we can induce the weak stimulus a couple of minutes after the strong stimulus, after the requirement of RAS MAP kinase activity in the dendritic spine that receives the strong stimulus. And we could then perhaps have time to intervene with LTP induction on the weakly stimulated spine using pharmacological methods. And that's what we did. So here's the experiment that we performed. Provided a strong LTP stimulus at time equals 0 and the subthreshold weak stimulus 5 minutes later. And in between, at 3 minutes, we interfered with the RAP-MAP kinase pathway by using a MEK inhibitor. We find is that in the absence of MEK inhibitor, even after 5 minutes for nearby spines, we can still evoke a good, robust long-term potentiation with the weak stimulus in the neighborhood of the spine that received the strong stimulus. When we now put the MEK inhibitor on at time equals 3 minutes, it has almost no effect on the amplitude of the LTP for the spine that received the strong stimulus, but the MEK inhibitor dramatically reduces the amplitude of long-term potentiation in the dendritic spine that receives the weak stimulus. This really shows that the spread of RAS activity is involved in reducing, is critical for reducing the threshold for LTP induction over this 10 micrometer stretch of a dendrite. So what I've shown you is that plasticity at one synapse facilitates induction of plasticity at neighboring synapses. So neighboring synapses are coupled by diffusible activated factors, including RAS, the likely other players. And that coupling, that crosstalk of synapses really extends over about 10 micrometers of dendritic territory, involving 10 to 20 synapses. And one might speculate what could be the biological functions of this coupling between nearby synapses. And this is only speculation, but this kind of mechanism could be a learning rule that binds semantically related inputs within the dendritic arbors of individual target neurons. So it's been shown using cellular physiology experiments that neurons that make synapses nearby within the dendritic arbors of the target neurons, when they're coactive, have a much larger impact on the postsynaptic cell than if their synapses are wide apart in the dendritic arbors. And that's because of strong nonlinear supralinear interactions of these synapses within the dendritic arbors. And of course, it would be quite powerful if then neurons that process or that encode related input would place their synapses nearby on their target neurons. And so the kinds of mechanisms that crosstalk in synaptic plasticity provides could actually be a learning rule to place these synapses correctly. So imagine here you have two neurons that have weak synapses on one dendritic branch interspersed with some other neurons from another group of neurons that really encode unrelated information. Now imagine that neuron 1 and 2 have firing patterns that are coactive over this time scale of a couple of minutes. So neuron 1 might induce synaptic plasticity at one synapse, and then if neuron 2 fires sometime later, it might also strengthen those synapses. And so it would bind the inputs of these neurons together at that dendritic location and perhaps compete away the input from another group of neurons that doesn't show correlated activity. And in this fashion, neurons that have correlated activity in time that might be semantically related, for example, neurons with related place fields would occupy and target the same region of the dendritic branch. So in other words, that semantically related inputs would now be targeting small regions of dendrite and then would be effective in driving more complex percepts. So for example here, one neuron in this cartoon, of course, that says something about the color of orange, another one, ball, would drive that neuron then to perceive or to be active with the perception of a basketball. So I'd like to finally acknowledge my collaborators in the projects that I've shown you. Most of the data that I had time to talk about were collected by Ryohei Yasuda and Chris Harvey. Ryohei is now Professor at Duke, Chris Harvey a postdoc at Princeton. I've had time to show you bits and pieces from experiments from other postdocs in the lab, in particular Anthony Holtmart, Linda Wilbrecht, Graham Knott, and Thomas Ertner.